Yes, good evening, and thank you for the kind introduction. So today, let's see, get this to advance. There we go. This is a brief outline of my presentation for today. We'll begin with an introduction to the structure and function of the microbiome and its temporal development. Then we'll take a little bit of a deeper dive into the factors affecting the development of the microbiota. I will then provide a brief overview of the gut microbiota impacts on host immunity and allergy development, and then end with some take-home messages. So oftentimes the terms microbiota and microbiome are used interchangeably. However, the microbiota refers to the collection of microorganisms that live on or in our body. Most of those are in our gastrointestinal tract, but we basically have bacteria on all surfaces of the body. And it refers to bacteria, but fungi, yeast, viruses, archaea, and protozoa as well. When we talk about the microbiome, we're referring more specifically to the genomes of the microbiota. And we know that there's about 100 times more microbial genes than the human genome. So we know that the products of the microbiota, what we oftentimes are referring to postbiotics, are very important to signaling to the host and in normal development. So we know that the microbiota is essential for organ and system development, also contributing to host nutrition by breaking down food components, synthesizing amino acids, and vitamins. Also resistance to pathogens. Much of this research has come from germ-free animals, but there's good clinical corollaries as well. So now turning to the infant, when we look at the infant gut microbiota, it develops in a sequential stepwise fashion. So the first colonizers are actually facultative and aerotolerant genera, which work to reduce the oxygen levels in the intestine. And that creates an environment in which more obligate anaerobes, such as bifidobacterium, can be established. So I like to refer to this early system as seeding, feeding, and weeding. And so the vaginal microbiota and particularly the maternal fecal microbiota provide some of the initial seeding. The infant is also exposed to environmental microbiota, which they can acquire from the hospital or where they're delivered, and other aspects in the environment. Once that seeding is done, then dietary components, particularly oligosaccharides, can come in and feed and subsequently shape the intestinal microbiota composition. And lastly, antibiotics, which we'll discuss a little bit today, can disrupt this ecosystem by non-selective weeding of commensal bacteria. I'm going to briefly talk about the TEDdy study, which is really an elegant study that probably has the largest numbers of samples that have been analyzed to date. So this was a study conducted by Christopher Stewart and colleagues. They obtained fecal samples from infants who were enrolled in the TEDdy, or the Environmental Determinants of Diabetes in the Young study. There were 903 children, and fecal samples were collected between 3 and 46 months of age. So I want to make a point that although this is very extensive and very informative, samples in the early postpartum period are missing in this cohort. They then applied what we call 16S ribosomal RNA sequencing, and that basically tells us who's there. These are tags that are unique to bacteria and some archaea. And then they also did metagenomic sequencing, which is whole genome sequencing. So this is primarily used to determine functional potential, but you can also derive taxonomy from metagenomic sequences, so you can determine who's there as well. So a little bit of an overview of the study and the results. So this top panel shows the feeding mode, and I've indicated which at the first time point at 3 months, which infants were human milk fed. So they had some that were getting formula alone or formula plus solid. This little purple area is actually the only infants who were getting breastfed only or exclusive breastfeeding, and the others were receiving breast milk along with formula or solid. So it was not a really exclusive breastfed cohort. And then they followed them across time. So these are months in age, so 10, 20, 30, and 40. And as has been shown in many other studies, we see this increase in the diversity. So that is telling you who the numbers are, the diversity of the bacteria that are there. And this gives an indicator of these operational taxonomic units, or the richness or the numbers of bacteria. So particularly in this early phase, somewhat plateauing here between 20 and 30 months of age, we see rapid increasing in both diversity and richness. These bottom two panels show the phyla. So this is the highest level denomination for the bacteria. And you can see, for example, that during this time that the firmicutes are increasing, the actinobacteria are decreasing, and proteobacteria are also decreasing. So we see changes in the overall phyla, and these are accompanied also by changes in the functional capacity of this microbiota. As they, particularly as solids begin to be introduced, we start to see changes in the bacteria and then in their functional components. So this is a little bit overwhelming at first, so let me walk you through this. But this was a really interesting approach that they were able to take, where they used this Dirichlet multinomial mixtures, which is a clustering program. And so basically they were able to put their sequences in, and not only could they identify who was there, so you see the bifidobacteria here at the top, and then some Bacteroides and others, but they could bin them into these 18 bins of when they were most predominant. And so it goes from 1 to 18. And so you can see bifidobacteria very predominant in these first 3 bins, but persisting really across until these later bins. You can start to see the emergence of some of the Bacteroides, and here are some of the more adult type Calicibacterium, Akkermansia, and Ruminaceti. So then they looked at these bins, and they, across the top, just to orient you, these are the time points. So 3 to 6 months, 7 to 10 months, 11 to 14, et cetera. The numbers of infants, so you can see that over time, the numbers of infants went down. But from the various clusters or bins, they were able to divide the establishment of the microbiome into various phases. So the first they deemed the developmental phase. So this is showing that there's really not a lot of variety here in this early time point, and the larger the circles are, then the more predominant the bacteria. So you can see some change in this. They were in this phase from 3 to 14 months. Then they moved into a transitional phase, which were the bins 8 to 12. This was between 15 and 30 months. And you start to see this movement from the developmental phase, and then eventually movement into the what they call the stable phase, which was predominantly here after 31 to 36 months. So this is really after 3 years of age. This is in that 1.5 to 2 years of age. So basically, this is a very elegant way, I think, of showing the data and showing how these infants are undergoing this kind of succession of their microbiota over time. And then using that data, they were also able to say, well, which bacteria are most discriminatory at each time point? So the reds are the more than 30 months, the blues are the 3 to 15, and then the greens. So the bacteria that actually was most age discriminatory was this Anorostipes, and I'll come back and talk about that a little bit later. And that was observed in the 15 to 30 months of age. But you can see in the young infants, you can see Bifidobacterium, Escherichiae, Lactoclostridium, and others. So this is just a really nice way for us to begin to understand which bacteria seem to be contributing at each age to being able to discriminate younger versus older infants. And this is just another way of seeing these are the bacteria that are more predominant in early ages. You start to see these come in between 15 and 30, and then these greater than 30. It's interesting to note that a lot of these still are persisting, whereas many of these early infant samples, our bacteria really are disappearing from the population. So this is the first poll question that we have for you. So which statement does not describe the development of the infant microbiota? So bifidobacterial species predominate in the first year. It occurs sequentially. Its composition is stable by 1 year of age, and bacterial diversity increases over time.
So for now, we have 62% that report its composition is stable by 1 year of age.
Okay, that's right. I think this TEDdy study and some of the others I will show that we're still undergoing quite a bit of change in those up to 3 years and potentially beyond. Okay, so now let's close the poll and we'll go on to the next section, which will be taking a little bit of a deeper dive into what are these factors that are affecting the temporal development of the microbiota. And I'll talk about mode of delivery, antibiotic exposure, and early life nutrition. But I really want to make the point that these factors are interacting with each other and cannot really be viewed as single factors. So this is a very nice figure that summarizes the various factors impacting the microbiome throughout life. And you can see this to depict again throughout even to mid-adulthood on this idea of increased abundance and diversity. Studies have shown that even during gestation, maternal diet, BMI, gestational diabetes, and excessive weight gain can impact the microbiome and subsequently impact the microbiome of the infant as well. Then at birth, the age in which the infants are delivered, how they're delivered, and whether they were preterm and in the NICU can all impact the microbiota. Here in bold, whether that infant's breast fed or formula fed, introduction of solids, et cetera. And then it's kind of important to note this big white space here. And this is a real gap in the literature. And as you can see, I said, okay, the TEDdy study studied till, close to 4, but then after that, we really don't have a lot of data. You can see there's a lot of question marks. So I think that we need to do a lot more research from beyond infancy and filling into adulthood. So originally they only had antibiotics affecting the microbiome in infancy, but I've added really antibiotics can cause dysbiosis throughout the lifespan. So now going back, looking a little bit, focusing more on infancy. So this is another figure from the Stewart paper. And then looking at these ages across the top, he divided the potential covariates into maternal, perinatal, gender, diet, supplement, environment, and clinic. So you can see the color coding here. And the asterisk shows which are significant covariates. So you can see that in the first 3 to 6 months, that birth mode is playing a role, that breast milk is playing the most dominant role, and that persists up until this 11 to 14 months of age. You can also see that other supplements as well as environment are playing a role. And some of these, become less significant over time and others are persisting. So geographical location, for example, persists over time. So now looking at antibiotic exposure and microbiome development. So antibiotics, as we all know, can exert a strong influence on the microbiota. And in the infant, it particularly affects the bifido. And studies have shown that antibiotic use in the first year is associated with an increased risk of allergy, asthma, eczema, wheeze, and atopy, and inflammatory bowel disease. So the hypothesis is that antibiotic-induced dysbiosis may be having impacts on that infant immune development. And infants can be exposed various ways, but it can be through indirect exposure, most often with intrapartum antibiotics during cesarean section. or direct administration. And this has been confounding because since antibiotic use is standard care, it was difficult to tease apart, well, are the differences with cesarean section due to antibiotic exposure or due to C-section? So this was a recent study in Nature Communications out of the Netherlands, and they did, again, a simple but elegant study in which they simply delayed the routine antibiotic administration until after cord clamping. And they compared vaginal and cesarean infants. So really they were able to uncouple the vaginal or the cesarean delivery from antibiotic exposure. So this shows the vaginal delivered infants' fecal microbiome from day 1 to month to a year of age. And again, simply just looking at the bars, you can see that there's large differences, and particularly in this early period of time. So if we look at cesarean infants, derived infants, their gut microbiota appears less stable. The colonization with bifidobacterium is delayed. So if you look particularly like 1 and 2 weeks, if you compare these bars, we see a higher abundance of potential pathogens such as Klebsiella and Enterococcus. And they followed these infants to a year, and they found that the relative abundance of these two at 1 week of age was associated with a higher risk of respiratory infections in the first year of life. So there are potential health impacts of differences. And so the take-home message is that even, removing antibiotic exposure, cesarean delivery does impact the microbiota, and it can persist. So then looking at antibiotic exposure directly to the child, we see this was a study where they took antibiotic naive infants who were hospitalized for RSV. They were even either given amoxicillin or macrolide, and they did sampling in the hospital, but then also followed out at 1, 3, and 6 months. And you can see a single course of antibiotic reduced both diversity and richness. It reduced the abundance of the bifidobacteria, which is shown here in red, increased enterobacteria and Clostridium. And even at a year, there was antibiotic exposure was associated with increased Clostridium. So this is the Clostridium, the Raceae and Lactobacillus and Ruminococcus. So the take-home message is that even a single dose of antibiotic in early life can have long-term implications and dysbiosis. So now what about feeding mode? So as we've mentioned before, that it's the primary determinant of microbiome composition, but it interacts with mode of delivery. So this is a study where these infants were all vaginally delivered, but then they were either breast or formula fed. So again, you can see that the vaginally delivered infants had more of the actinobacteria with this lighter blue, whereas there was more Bacteroides here in the formula fed. So why is this happening? And Dr. Bode will talk more about this, but we know, for example, that human milk contains many bioactive components that can either work to suppress or promote the growth of specific taxa. So again, human milk, it's not just the human milk oligosaccharides, which we know are certainly important, but there's many other factors. And it could, it's been proposed that in the early time postpartum, that components like secretory IgA and lactoferrin are actually suppressing the growth of Enterobacteriaceae, and then HMOs are helping to increase the abundance of specific bifidobacteria. I'm just going to quickly show you some data from my laboratory, because we know that many infants are what we call combination fed. They're not exclusively breastfed, or they're not exclusively formula fed. And so this is a longitudinal cohort that we're following. And I'm going to kind of focus on the difference between these. So we have these combination fed infants who on average were getting about half human milk with a broad range from 8 to 95%. And this shows how this is an overall principal coordinates analysis of the infants. So these are the formula fed. You can see they cluster quite tightly compared to the breastfed, and these groups are significantly different. But what really surprised us is that the combination-fed infants were actually clustering more closely with the exclusively formula-fed than the exclusively breastfed. But again, we're showing a broad variation, likely again due to the fact that we had from 8 to 95% human milk being consumed. When you look at this again, and this is just for you to basically look at the breastfed versus the combined and formula-fed, and just looking at this, you can see both at the phyla, genera, and species level that the combined and formula-fed infants are more similar to each other than they are to the exclusively breastfed. And this shows metabolites. So we did fecal metabolomics, and the same story plays out, but it's even more clear where the formula feds and the combination feds are clustering more tightly than the breastfeds. So this is an area of active research where we're really looking at, what are the components and what are the functional implications of the fact that the breastfed and infants are distinctly different from formula and the combined fed. So another poll question for you. So which factors influence the development of the microbiota? Route of delivery, diet, environment, antibiotics, or all of the above? Okay, so where are we with the numbers?
We're at 93%, all of the above.
Very good. Okay, so let's go to this last part. And again, this is such a complex topic that I'm just going to give you a very quick overview, being cognizant of my time. So when we look, this is a very hot area of research. As I mentioned before, as we see dysbiosis being associated with many clinical outcomes that are related to the immune system, not only allergy, but asthma, wheeze, and even inflammatory bowel diseases. So when we look at prenatally, it's important to note that we often say they're immunologically immature, but most of the secondary lymphoid structures, they're there. They just have not had extensive exposure to the microbiome. So we have these M cells, which overlay the Peyer's patches, which I'll show our sampling, dendritic cells, the T and B cells in the mesenteric lymph nodes. And so in a normal situation, in a homeostatic situation, we are educating this developing immune system, and with commensal bacteria. So these M cells, or microfold cells, are sampling luminal antigens through endocytosis. These dendritic cells are then presenting antigens in a T cell dependent manner to promote B cells. And these B cells are secreting secretory IgA. So everything is happy. We have a nice mucosal barrier and protection. The dendritic cells can also sample bacteria. The MAMPs present those to T cells and induce T cell proliferation and differentiation. And so you're seeing Th2 and Tregs and secretion of IL-10 and TGF-beta. So again, in a homeostasis, you have this crosstalk continually happening between the commensal microbes and the microbiota, and that is good to commensal microbes in the infant to induce tolerance and avoiding autoimmunity and allergy by these cytokines that are being secreted. So in a state of dysbiosis, where you can have pathogens or you can actually have tissue damage, we then go to a scenario where these pathogenic MAPs are sensed, causing the secretion of pro-inflammatory cytokines, which educate effector T cells, which then put them down a different pathway. So instead of Th2 and Tregs, they go to Th1 and Th17s. And basically then we're also seeing that B cell maturation in the Peyer's patches is inducing IgG instead of IgA. So simply by having the pathogens and affecting the mucosal barrier can put that infant towards a much more pro-inflammatory state. So kind of quickly go through what do we know right now. So there's emerging data that this early colonization is associated with food sensitization and persistence. And I'm going to just point out, so the Consortium for Food Allergy is finding that Clostridium were enriched in the gut microbiota of infants whose cow milk allergy resolved by 8 years compared to those that persisted. So here we're looking at persistence. Work with the Megan Azad published from the child cohort in Canada found that each quartile of microbial richness at 3 months was associated with a 55% reduced risk of food sensitization by a year, but there was no association with that infant's microbiota in a year. So really this idea of earlier microbiota likely programming. And then again, enterobacteria were overrepresented, and these include Clostridial species. Another study found that there was a number of OTUs that differed, including several species of Clostridiales. So the kind of take home here, you'll see Clostridium is coming up as a protective bacteria in these studies. And then just to finish up, this has actually then gone into germ-free animals who are susceptible to allergy and fecal microbial transplants. And what this group showed that a microbiota from healthy control subjects, but not subjects with food allergy, protected these recipient mice from developing food allergy when they were challenged. And then they showed that a cocktail of 6 Clostridiales protected the germ-free mice. So this was a full fecal transplant, and these are specific bacteria, whereas proteobacteria did not. So again, just pointing to some aspect of these Clostridiales. And this was a study from Kathy Nagler's group at University of Chicago, which got a lot of press. And so she took fecal microbial transplants from infants fed a milk formula versus those infants with a cow milk allergy that were being fed a hydrolyzed formula. She reasoned that they were likely being fed different formulas. They were put into the mice, and then they were sensitized with beta-lactoglobulin. And the mice with the cow milk allergy transplant had symptoms to the lactoglobulin, but those who had the healthy donors did not. So it was really showing that this cow milk allergy microbiome was carrying that phenotype. And then they were able to directly identify an Anostypes caseae, and which again, a Clostridial species that they showed induced regulatory genes. But then when they gave that along with a milk formula, it was able to protect mice from milk allergy. So in summary, that I hope you are coming away with that the early postnatal period is critically important to establishing the gut microbiota, that route of delivery, diet, and antibiotics can interact, and that if this is not done ideally, if we have dysbiosis, emerging evidence is suggesting that these microbial exposures in very early immune development can play a role in the natural history of allergy. And so this is particularly, studies are showing it's that microbial dysbiosis in the first 3 to 6 months of life. And then again, the more mechanistic studies that are showing that a food allergy microbiome can transmit susceptibility, but there are others that can inhibit, and they've gone on to really begin to tease out the molecular mechanisms. So thank you.
All right, thank you very much. Can you see my slides, and can you hear me okay?
Yes, we can.
Okay, I'll take that as a yes.
Yes, we can.
Okay, thank you. Okay, so we talked a lot about how important the gut microbiome is, especially early on for lifelong health and development. And I'm really going to dig into what is it that is in human milk. And you've seen this slide here from Chris Stewart's paper in Nature, where specifically highlighted this large impact of breast milk on the developing gut microbiome. So for us, the question is, what is in breast milk, what is in human milk that really drives this major variation in gut microbiome that then has long-lasting effects? So if we lift the curtain here and see what is in human milk, and you've seen already there's so many different bioactive components, I'll just focus on the macromolecules here. There's protein, fat, lactose, and then there is oligosaccharides, 5 to 15 gram per liters in human milk, which often exceeds the amount of total protein. If we compare that to bovine milk, there's about 100 to 1,000-fold less oligosaccharides in bovine milk than there is in human milk, and some other species have different oligosaccharides, different amounts of oligosaccharides, but really human milk stands out as being fairly unique. And as a consequence, infant formula is not having most of these human milk oligosaccharides that we have in breast milk. So there's a huge difference. And let's take a quick look at what these oligosaccharides are. And I'm really just focusing in the 20 minutes that I have on human milk oligosaccharides. There's many, many other components as well, lactoferrin and antibodies and secretory IgA. So just focus on the oligosaccharides because they're really currently the hot topic in the field. Oligosaccharide means a few sugars. So if we look at that, what does this really mean? This is the sugar you know from what you have in your food, in your coffee in front of you right now. We don't really want to have that simple sugar in our diet in the early stages. I'll show you that the human milk oligosaccharides are way more complex structurally than what we have in table sugar that you put in your coffee and tea. So to reduce some of that complexity here, we make this fairly easy and give each building block that we have for the oligosaccharides a little symbol here, put them together that way to make it easier to talk about it so we don't have to pull up the chemical structure each time. And there's really only 5 building blocks for human milk oligosaccharides: glucose and galactose, which together makes lactose. And then we have N-acetylglucosamine, fucose, and sialic acid. So this is almost like Lego bricks. Depending on how you put those building blocks together, you get different oligosaccharides, or for the Lego analogy, you get either a little car or you get a little plane, and you'll agree that little cars and little planes do different things, and it's the same thing with oligosaccharides. So the oligosaccharide structure determines its function. Different oligosaccharides do different things. And to keep this very simple, I'm going to show you in the spot on the top here, the blueprint of human milk oligosaccharides, so how they are put together, and then in the white space, some of the examples of what oligosaccharides we have. So all human milk oligosaccharides start with lactose at the reducing end, and then they get extended with disaccharide units. They can be branched, they can be further elongated, so the backbone alone gets fairly complicated. And then we can add fucose to these structures, either to lactose alone, and we get 2 and 3 fucosyl lactose, or to the extended backbones in different linkages. So you see there's really a whole array of oligosaccharides building up here. And then we can also add sialic acid in different linkages, again, either to lactose alone or to the growing chains. And you have an arsenal of about 150 to 200 different oligosaccharides identified in human milk so far. And it's very important to keep in mind that every mom makes a different set and different concentrations of human milk oligosaccharides. Here's a study that we did with the Canadian child cohort. You see there's about 1,200 milk samples analyzed and depicted here on the graph. And every color is a different oligosaccharide. And you can see over the spread here from left to right, how the total concentration is very different, but also how the colors are different between the different samples. So really every mom has almost like a thumbprint of different oligosaccharides here. That is remarkably constant over a short period of time, so over a week. And even if you look at a higher resolution, you look at from hour to hour, the oligosaccharide composition is remarkably constant, but very different from mom to mom. That does change over the course of lactation over a longer period, so from 1 month to 6 months to 12 months, but over a short period of time, it's remarkably constant. Here's another way to look at the variety of human milk oligosaccharides. This is a principal component analysis of oligosaccharide composition in about 10,000 milk samples. And you see in this 3-dimensional space, the closer those dots are together, that the more similar the oligosaccharide composition is. And the further apart the oligosaccharides, the dots are, the more dissimilar the oligosaccharide composition. And this is really collected from multiple different areas around the world. So here comes the first poll question. I can't see what you're answering there, so I give you the answers here. So we'll state all that are correct. HMOs are the 3rd most abundant solid component of human milk. HMOs are complex sugars. So far, 150 to 200 different HMOs have been identified. Bovine milk contains more HMOs than human milk, and every person synthesizes the same set and amount of HMOs. Okay, what are the answers?
So mainly it is answer A, B, and C that were picked.
And that is exactly correct. So it's true that it's the third most abundant component. They're complex sugars, and there's about 150 to 200 different oligosaccharides identified. There's more in human milk than in bovine milk, and it's very different from human to human. Okay, let's move on. So what are the current questions? One is, what actually drives this variation from mom to mom? What are the maternal drivers that impact oligosaccharide composition? And then, of course, what do these oligosaccharides do to impact infant health and long-term development? And last but not least, we're also looking at what can these oligosaccharides do and can we learn from what they do in the maternal infant space to then develop therapeutics for people of all ages. So because we have very limited time, I'm only going to focus on infant health and development. How do we study human milk oligosaccharides? Where we use the entire bandwidth coming from in silico, in vitro, ex vivo models, then going in vivo models, doing association studies on large cohorts, and then eventually doing intervention studies where we can test the effect of individual oligosaccharides or mixtures thereof. And all that is not just done in our center here at UCSD, but we really use a network of collaborators from around the world to answer all of these questions and use all of these aspects of the pipeline. So let's focus on one specific disease area, just one slide to show you how we work on oligosaccharides and what's needed to really get to some answers there. And this is in the context of necrotizing enterocolitis, which is a disease that affects very low birth weight infants, preterm infants, and affects about 5% of them. On the other hand, we know that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop this disease. And our question was like, what is in human milk that prevents this devastating disease in the preterm infant? Is it potentially HMOs? So the way we studied this, and this is one slide, is about 12, 13 years of work, and we highlighted that in a review paper in 2018, where we isolated first human milk oligosaccharides from pooled human donor milk to get the entire garden variety of different oligosaccharides isolated, and then tested that in an in vivo model of this disease. We then found that, yes, there is a very strong effect on survival and on pathology, and wanted to know, well, which oligosaccharide is it? Is it all of the 150, or is it one specific one? So we used multidimensional chromatography to break this down into individual oligosaccharides in the end, and we found that the effective oligosaccharide looks like this. It's called DSLNT. We then developed the chemical space around that, but also wanted to know, does this translate to humans? So then we set up a mother-infant cohort with moms that had very low birth weight infants, and we collected milk samples every second day of what went into those infants over the first 28 days of life. We then analyzed oligosaccharides and looked, are there any oligosaccharides different in the milk that went into infants that later developed necrotizing enterocolitis? And indeed, we found that there is a huge difference. Those infants that developed this disease had less of the protective oligosaccharide DSLNT than those that did not develop the disease. So it matched very nicely with the preclinical data. And we've shown this in a third cohort now that's currently under review after revision, also in gut. So it should be coming out in the next few days. So that really leads us to move into clinical studies and also to stay in the animal model to look at the mechanism. So one oligosaccharide has an effect here. So we can now say that, yes, HMOs are important to protect from NEC, and it is one specific HMO called DSLNT. But really, this is just one example. So we're looking in the preterm space at the prevention and treatment of NEC and sepsis, but we're also beyond that looking at overweight and obesity. Are there oligosaccharides that help us prevent overweight and obesity, and can we use that for treatments afterwards? The same is true for diarrhea and infections. The same is true for cardiovascular disease. Very cool data showing that if oligosaccharides, specific oligosaccharides, are there in early life, the risk for cardiovascular disease is lower, and we can use these oligosaccharides as treatment for people of all ages. And then we also work in the cognition space here when it comes to early life potential and later life treatment. Now, that's all great. That's all very ambitious goals and projects, but how does this actually work on a mechanistic level, and what is that to do with microbes? First, before we get into that, very important to know that oligosaccharides are very resistant to the low pH in the gut. Do not get broken down by pancreatic and Bode enzymes. About 1% is absorbed and makes it intact into the systemic circulation, and the rest is degraded in the colon by bacteria or is excreted intact with the feces. Here's a schematic that really shows that interaction between oligosaccharides on the top, good bacteria in green on the left, potential pathogens in purple on the right, and then the intestinal epithelial cells and the underlying immune cells on the bottom. So for example, we know that oligosaccharides are prebiotics, so they foster certain potentially beneficial bacteria. And we've done some work here with Dr. Carsten Zengler at UCSD, where we looked at structure-specific effects of human milk oligosaccharides on bacterial communities that we extracted from infant feces. So it's really ex vivo studies. And if we just look at growth, we see that over the first 48 hours here, if we feed human milk oligosaccharides, these bacterial communities grow very nicely. Also, if you feed them individual oligosaccharides like 2FL and LNT, you see a nice growth curve. So they use the oligosaccharides as substrate. We repeated this experiment. A good scientist does experiments multiple times to see what's going to happen. And you see the effects are very similar. They grow on pooled oligosaccharides, LNT, but they do not grow on 2-fucosyllactose for some strange reason. And we see that 2-fucosyllactose is not utilized by these bacteria. And you see this here on the very right. So for us, the question was, why is the community on the top metabolizing all oligosaccharides, on the bottom only select oligosaccharides? And we found out this first mother here is a secretor, meaning this mom A makes a lot of 2-fucosyllactose. So that means that her infant and the infant's gut microbiome were exposed to 2FL prior. So those bacteria already knew what to do with 2-fucosyllactose. On the other hand, mom B is a non-secretor, cannot make 2-fucosyllactose. The infant and the infant's gut microbiome had never seen 2FL and did not know what to do with it, so they couldn't grow on it. To infants and the bacterial community in those infants, depending on what they were exposed to prior already. But this is really just looking at growth at this point, right? So this could be any bacteria growing, potentially bad bacteria as well. So we went a little bit deeper and did 16S sequencing on some of these samples. And again, the closer these dots in the three-dimensional space, the more similar the bacterial communities in these samples. So we start out here, we feed these cultures pooled human milk oligosaccharides, and the cultures move in the three-dimensional space, meaning that the composition changes slightly. And we see that that changes again after 48 hours. More importantly, if we feed them just LNT, you see the communities move in a completely different space in that three-dimensional space here. And if you feed them 2FL, the spot is very different as well. So in other words, yes, the growth of total bacteria might be very similar or identical, but the community structure, so what kind of bacteria are in that structure, are very different. So it really matters again, and it tells us again that structure, even on the prebiotic case here, determines function. Okay. So that is very important because when we now look at the development of pre, pro, and symbiotics, we can really take a look at what bacteria metabolize what oligosaccharides and what do they actually do with it. And I give you a few examples here real quick. What you see here is actually 2 chromatograms of human milk oligosaccharides. It looks like really just one in purple, but the black chromatogram is just underlaid under that, and they are almost identical. So that means that this is before and after use of probiotics, and the oligosaccharide composition is identical. In other words, these bacteria do not utilize common human milk oligosaccharides. Here's another example, and now you can see down on the bottom here that there is a black line popping up. So that is the oligosaccharide profile after exposure to these bacteria, and there is entire oligosaccharides diminished or even missing, including DSLNT. That is the oligosaccharide again that was protective in necrotizing enterocolitis. So, and then there's other oligosaccharides, and you can specifically see that multiple different oligosaccharides are utilized, almost all of them at the same, to the same extent. And then there's other spots popping up here, though this is the degradation products of these oligosaccharides. What we ideally want to have, especially now in the preterm space and necrotizing enterocolitis, is developing a probiotic together with a prebiotic where DS-LNT is not used. So here you can see this probiotic specifically uses LNT. And you can see that it's missing in this figure here. But it does not go after DS-LNT. It leaves it intact, and that's what we want. We want to have a probiotic that works as a probiotic, but leaves the prebiotic intact to do all the other magical things. So now, what are all the other magical things? We think that oligosaccharides are more than just food for bugs. We know that they serve as antimicrobials. So not only do they foster the growth of certain bacteria, but they actually inhibit the growth of other bacteria that we don't want. They also serve as anti-adhesives. And I'll show you what that means. Many bacteria need to attach to the intestinal epithelial cell surface in order to proliferate. And that attachment is very often mediated through lectins and interactions to the sugar molecules on the epithelial cells. That's very important. So the bacteria need to hold on. They need to be able then to proliferate, in some cases, to invade and cause disease. Now, in the presence of human milk oligosaccharide, it turns out that these HMOs mimic some of the structures of the epithelial cell glycocalyx, or the sugars on epithelial cells. Now, the bacteria bind to the human milk oligosaccharides, not to the epithelial cells, and then they get washed out without causing disease. So we've seen this here in an example with enteropathogenic E. coli. Others have shown this with Campylobacter. And you can show that it's not just adhesion that is blocked, but it's really lesion formation, so destruction of the epithelial cells that is blocked in the presence of human milk oligosaccharides, and that is significant. We've been able to translate this into an animal model and identified one specific oligosaccharide that is responsible for this effect. And we're able then to design a synthesis strategy to make this oligosaccharide available, retest it, and have an oligosaccharide now that can attack this specific pathogen. But then there's more. Oligosaccharides have direct effects on epithelial cells, which then can backfire into the intestine and change the microbial community that way as well. And the same is true with immune cells. Oligosaccharides can modulate immune cell responses, which then also backfires into the intestine and can change the microbial community. These are all local effects, but we know that 1% is absorbed, and that is a fairly high number. If you think about 15 grams per liter, 1% of that is substantial. So we can also have systemic effects on the immune system, on the brain, on many other organs within the infant. And we actually see that. So if we look at specific oligosaccharides, they are correlating with cytokine releases. We see for 3SL that there's a very strong effect on chronic macrophage inflammation. We've done a lot of work on leukocyte activation and extravasation and found specific oligosaccharides working on that. There is work on excuse me, on TH1, TH2 balance. And then Dr. Donovan has done some very nice studies there in piglets looking at cytokine releases and other cell types and immune markers. So these are all from in silico and in vivo models in animals, but we also have human cohort studies where we can link human milk oligosaccharides specifically to cow milk allergy, to food sensitization, and to allergic disease. And this paper just got accepted recently. So not only can we model this in vitro in tissue culture and animal models, but we then see that there is associations relevant in human cohort studies as well. And the next step would then be to test this in clinical studies. So really, what we think is we are here. We are at the tip of the iceberg. There's probably many more mechanisms of what oligosaccharides can do, immense opportunities to work in this space. So here comes the last multiple choice question of this webinar. Again, select all questions or all answers that are correct. HMOs can act as prebiotics, probiotics that utilize each and every HMO are the best. HMOs are more than just food for bugs. HMOs are digested by the infant, and intact HMOs can be found in the infant's urine. So I'll give you 30 seconds to answer. Since done with this webinar, maybe we can cut this short. And I'll give you the answers right away. The answers are HMOs can act as prebiotics. And no, ideally, we want to have probiotics that are targeted and do not use up all the oligosaccharides, because the oligosaccharides have a lot of other value as well. They are more than just food for bugs. They are not digested by the infant, and we find intact HMOs all the way in the infant's urine. Okay, so let's summarize this. Last slides, HMOs are the third most abundant solid component of human milk. They're a group of complex sugars. There are about 150 different ones. There is smaller ones, there is larger ones, there is ones that are silated and non-silated. There are some that are fucosylated and non-fucosylated. So it's really depending on what structure we are looking at. It's different molecules. HMO structure determines HMO function. They're not digested by the host. Some of them are absorbed. They are prebiotics, anti-adhesives, antimicrobials, and they can directly modulate epithelial cell and immune cell responses. They can affect outcomes like necrotizing enterocolitis, as I showed you as an example, but also infectious diseases and diarrhea, growth, neurodevelopment, immune system development and responses. And that is probably just the tip of the iceberg. And then I'm going to skip this part here. Sometimes we need specific oligosaccharides, sometimes you need a mixture, and I leave that for another day. Okay, I'll end this here and see if we have still time for questions.